Hello, bachcho. Let's see the question. So the question says restriction endonucleases are used in genetic engineering because. So हमें इन चार ऑप्शन में से रिस्ट्रिक्शन एंडोन्यूक्लियस को जेनेटिक इंजीनियरिंग में क्यों यूज किया जाता है ये आइडेंटिफाई करना है. So the key concept in this question is to first understand the use or the purpose of restriction enzymes or endonucleases in genetic engineering. Now, before understanding the use of restriction endonucleases, let us first understand what restriction endonucleases actually do. So, restriction endonucleases are firstly enzymes, and these enzymes act as nucleases, that is, they cleave the nucleic acid sequences, particularly DNA. So restriction endonucleases are responsible for the cleavage of DNA only at specific sites. So restriction endonucleases have a very interesting feature about them. That is, the restriction endonucleases only recognizes a particular sequence of DNA and then cleaves the DNA at a specific site only. So restriction endonucleases act like molecular scissors that can snip off DNA sequences into fragments at specific sites. So restriction endonucleases were first discovered in bacteria, and bacteria use these enzymes to fight against bacteriophages. So they protect themselves from bacteriophages. So these restriction enzymes present in bacterial cells are responsible for the recognition of specific sites in the viral DNA and then degrade or cleave the viral DNA into fragments, which results in the formation of non-functional viral genes, and therefore the bacteria is protected from the bacteriophages. Now, how can we use these restriction endonucleases for our own needs? Now, since restriction endonucleases can cleave DNA at a specific site, we can use or exploit this feature of restriction endonucleases for our own needs, where we can modify or manipulate the genes at specific or targeted sites as per our needs. Now, this feature is vastly used in a field of genetic engineering which is known as recombinant DNA technology. So the diagram given below represents the very basic steps of recombinant DNA technology and the use of restriction enzymes in recombinant DNA technology. So these are the restriction enzymes. Now these restriction enzymes can cleave the DNA at specific sites, and this is the plasmid and this is the foreign DNA or the gene of interest that we want to insert into the plasmid. So with the help of restriction endonucleases, we can remove the gene of interest. So this is the gene of interest. So we can remove this gene of interest from the DNA sequence by using restriction enzymes. So here we have the gene of interest after using the restriction enzyme. And in case of the plasmid, which is the vector, we can again use the same restriction enzyme and cleave off a particular sequence of DNA which is recognized by this restriction enzyme. So this restriction enzyme will cleave off this particular part of DNA and we'll have a plasmid, and this region will be empty for the insertion of the foreign DNA. So after the insertion of the foreign DNA into the site which was cleaved off by the restriction enzyme, we can then have the recombinant DNA. So in this way, we can get a recombinant DNA by using restriction enzymes. And a recombinant DNA is a DNA which has DNA from two different organisms. So that is a recombinant DNA. So this is how restriction enzymes play an important role in genetic engineering. Now, if we see the options given over here, then the first option says that restriction endonucleases are used in genetic engineering because they can degrade harmful proteins. So this is not the answer, because restriction endonucleases, they do not degrade proteins, but they degrade DNA. The second option says that they can join DNA fragments. Again, this is not true, because DNA restriction endonucleases do not join but cleave DNA segments. And the joining of DNA fragments is done by DNA ligase. So this is not the option. Now, the third option says that they can cut DNA at variable sites. Again, this will not be our answer, because restriction endonucleases do not cut DNA at variable sites, but they cut DNA at specific sites. Then the fourth point says that they can cut DNA at specific base sequences. So this will be the right option, because restriction endonucleases cut DNA at specific site, and this is what is exploited during recombinant DNA technology. So our final answer is option 4, that is they can cut DNA at specific sequences. So this is our final answer. Hope you understood this well. Best of luck. Thank you.